Hello, everyone, and welcome to this week's NeuroTools Webinar. Today, I have the privilege of introducing Dr. Sarala. If you'd like her last name, you can visit the new info webinar page. I will butcher the pronunciation if I say it here. She will be presenting Identifiers.org, Compact Identifier Resolution and Services. Sarala is the project lead for Identifiers.org. She envisions, implements, and manages the Identifiers.org system. She has a software engineering degree and a PhD in bioengineering. Furthermore, she's been involved in many different data integration and infrastructure projects over the past 8 years at the EMBL-EBI. She is actively involved in the Data Commons Pilot Phase Consortium on globally unique identifiers, otherwise known as GUIDs. The Elixir Interoperability Platform on BioSchemas and Identifiers, the Force 11 Data Citation Implementation Pilot on Identifiers, and the EU FREYA Project on Identifier Services. Just a little housekeeping. Please ensure that your microphone is muted during this webinar. If you have any questions, please feel free to type them into the group chat. I'll be monitoring that, and I will address them to Sarala after her presentation during a brief question and answer. Without further ado, I'd like to pass the microphone over to Sarala. Thank you so much for joining us today. We're very excited for this webinar.
Thanks, Devon, for the introduction. I'll try to keep the presentation for about 30 minutes, actually. It's 7 p.m. in the evening here, so it's time to go to the pub. But anyway. Let me dive straight into the main message that I want to get across. That is, I want to encourage everyone to annotate and cite their data using actionable compact identifiers. In my talk, I will introduce the compact identifiers and the services built around Identifiers.org to make these compact identifiers resolvable. So a bit about who we are. So I'm part of the EMBL-EBI. It's an international non-profit organization serving as the European hub for biological data services and research. And we are based in Cambridge, UK. And we are part of a bigger, larger organization. Our headquarters is in Heidelberg. The main organization is the European Molecular Biology Laboratory. And here in Hingston at the EBI, we have over 600 staff members from over 53 nations. So at the EBI, we host a large number of databases covering a wide range of biological entities. These include genes, protein sequences, genomes, molecular structures, pathways, ontologies, literature services, etc. So it's it's almost like NCBI, but probably a bit smaller scale. So in life sciences, we often use identifiers to identify entities uniquely. All our databases assign identifiers to identify the individual entities, and they're generally made actionable by embedding them in URLs. So for example, in my examples, the orange text is the identifier, and then the blue text is the resolver. And it is also common to have the same data collection served from multiple different physical locations. So sometimes providing better curation, better visualization, or different integration activities, etc. So, for example, taxonomy is hosted at NCBI as well as EBI. So, assigning identifiers is a great start, but the way we use it at the moment also poses some challenges. So multiple URLs for the same collection make the object unification challenging. And when you come to annotate these resources, which resource should you be using to annotate or cite the data? And also, how do we keep track of the changing URLs and the dead links? So to solve some of these issues, we have introduced this concept of compact identifiers. So the compact identifiers consist of two parts, the prefix, which indicates the assigning authority, and then accession number, which is the local identifier that's assigned by the local database. And in case of where we have multiple providers, we define an additional provider level prefix to identify the individual hosts. So Identifiers.org system provides a way to create and resolve these compact identifiers. We also promote fair data, and it is a community-driven project, so which means all the resources that we have added into our registry have been requested by a user and is also free to use. So Identifiers.org was first developed in 2005, and since then we've been supporting many different data integration efforts. So we are part of the Elixir infrastructure, and we are recognized as a stable Elixir resolution resource. For the next 3 years, starting from this year, we are involved in 2 projects in either side of Atlantic. One is called FREYA, which is EU-funded 3-year project on providing core persistent identifier services. And the other one is NIH Data Commons. And on that, we are part of the we are building globally unique identifier services. So in EU we call them persistent identifiers, and US it's called globally unique identifiers. So there's discussions about these terms as well. But anyway. I won't go into that detail. So we are very excited about these projects. So we have a registry over 600 manually curated life science data collections. So which means we are not harvesting this information automatically from different resources. We actually have a curator who maintains these maintain our data records and make sure all the links are working and the patterns work, etc. So for each of the collections we store some information about them. So for each of the collections we store a description, identify a pattern, different URI schemes, and also we assign a unique prefix to identify the data collection within the registry. And we also store the different resources that we can get more information about this resource. So for each of these resources, we store more information, such as the institutional details, some description, the access URL, and also the different formats that the data is available in. And we also record whether it's considered as a primary resource within the community. And we also have a tool that checks the uptime of these services, and we use this information during our resolution time. So And for each of these resources, we also assign a provider code, and this provider code combined with the unique prefix identify individual resources uniquely within our registry. So Identifiers.org resolver uses all this information stored in the registry to resolve the compact identifiers. To support this, we maintain the URL changes and server downtimes. So for example, if you have taxonomy :9606 and you pass it into Identifiers.org resolver, most likely you will be redirected to NCBI, because we record this as the primary resource, and also NCBI resources have a high uptime values. And if you want to access individual resources, you could also force our resolver by adding the provider code. So for example, if you want to go to OLS, then you go OLS/taxonomy :9606. So OLS is the provider code to identify ontology lookup service. And it is important to note that Identifiers.org system doesn't store the individual data entities. Instead, it maintains up-to-date data access patterns and the relevant metadata of resource providers. So in life sciences, it's a common practice to cite data entities using plain text, for example, protein data bank 2GC4. And in general, this is people in the field can understand it, but it's outside the community, it's really hard to understand. I mean, it's hard to find out where this information is stored or coming from. So what we are aiming for is to use Identifiers.org combined with compact identifiers to support a shorter but machine-readable, persistent, and less ambiguous citation. So the idea is you have PDB :2GC4, so PDB to identify the protein data bank, 2GC4 is the local identifier, accession number, and then if you pass it to Identifiers.org, you will be redirected to the actual content. So Identifiers.org system is built as a generic system. So this has enabled us to extend our system beyond the biomedicine. So for example, we could also resolve DOIs, ARCs, ORCIDs, etc. So if you have a resource that we already don't, if we already don't have it in our registry, then what you need to do is fill in a prefix request form, and that has all the information that we require to store to support resolution for your data resources, and we could assign a prefix for your resource. So it's in some cases people fill in everything, and it's easier to add it to our registry. If the form is not filled in properly, then it takes a bit longer. So if anyone wants their resource quickly in the Identifiers.org registry, then it will be good if you can fill in the whole form. We also provide sort of an advanced search for find valid prefixes, identifiers, and providers, and also validate compact identifiers. And we also have different web services that you can use to access the content in the registry programmatically. And we also provide the data in the registry as a XML download and a YAML file as well. Because we store different URI schemes in the registry, we expose this information as a virtual SPARQL endpoint. So the idea is if you have different RDF resources, which uses different URL schemes, then you can use Identifiers.org virtual SPARQL endpoint to do these federated queries without having to do string manipulation in your SPARQL query. And we also support we enable users to extract metadata from the landing pages. So we are part of this effort called BioSchemas. So with the BioSchemas effort is about encouraging data repositories to add the metadata in schema.org, JSON-LD on their landing pages, and services like us can extract this metadata. And the wider idea is the larger search engines can index this information. But for identifiers.org, we could, if everybody does this consistently and you pass in a compact identifier to our metadata endpoint, then we are able to scroll the landing page and get metadata. So this is ongoing work. So at the moment, if you have the if you have the metadata on the landing page as schema.org JSON-LD, and then we are able to extract this information. We are also working closely together with N2T Resolver that's based in California Digital Library. So the idea is that we both use the same prefixes and the formats to support resolution. So if you have a compact identifier, then you could pass it to identifiers.org or N2T to resolve the compact identifier. So this was initially started in 2016, and we have publication on this, which is just been accepted by Scientific Data, and hopefully will be available properly online. But you can view a copy of this in bioRxiv. And we also have a MOU between CDL and EBI for providing ongoing support for this work. And this was actually started as part of Fourth 11 identifiers work group. So it has been very productive from that end. And this is how our future's looking, basically. The idea is to move our infrastructure into the cloud as part of Data Commons pilot phase consortium work. At the moment, our infrastructure is running on production servers in the data centers maintained by EBI. And what we want to be able to do is move this into the cloud to provide better support for latency and availability. So for example, if you run a query from NCPI, it'll come all the way to UK for resolution. So what we are planning is we will have two layers. One layer is the core data set and services, and then the distributed layer. So the idea is the distributed layer will get the information that's needed for redirection, and the registry and metadata, and will be hosted in multiple different clouds and different locations. So the queries will be answered fairly quickly and depending on the geographical location. And also this way, the newer infrastructure will be quite decoupled, so you will be able to run these distributed services wherever you want, and also connect to our core data sets to extract the data. So this is ongoing work. We have some initial work done on this. Hopefully we'll be able to release them in the next couple of months. So coming back to my take-home message, the main thing, it's to annotate and cite your data using actionable, compact identifiers. Please do get involved. You can follow us on Twitter, and you can also request prefixes through our request form. And all our code is in GitHub. You can access all the information there. And we also have our new bits of the new infrastructure in Docker Hub, so you can try instantiate sort of running a Docker container for the resolver and see how that works. And I would like to finally thank everybody who's been working on this work. So Manuel for the sort of infrastructure side of things, and John and Greg for all the harmonization work, and Nick for all the curation work that's been done, and Tim for leading the identifiers working group within Force 11, and also Rafa for all the schema.org related work within identifiers.org. And I would also like to thank all the funding for us to be able to continue with this work. So thank you for that. So that's all from my end.
Thank you so
much. Have any questions?
I do have one, actually. I was wondering if you could elaborate on SPARQL a little bit for people that might have less experience with queries.
Less experience with SPARQL queries or queries?
Yes.
Okay, so so we have at the EBI we have RDF store, and we have sort of we say, okay, for taxonomy we use this URL, and for other things we use identifiers.org. So we have a set of rules that everybody at EBI follows, so you can link it all up. But then you have the resources like Bio2RDF or even Uniprot endpoint. If you want to connect these endpoints together, you end up with, you have different URI schemes that's being used in different RDF stores. So what our virtual SPARQL endpoint does is, if you send a gene ontology URL in Perl, then you can get all the gene ontology different URI schemes that we have in our registry for gene ontology. So this way you don't have to do the string manipulation. Does that make sense? Let me go to the slide again.
That makes sense to me. Do you think you could point us to a URL where that is written up? There it is.
Yeah.
Perfect.
So for example, I think this is the EBI RDF store uses Perl for identifying gene ontology. And Bio2RDF uses Bio2RDF URLs. So in our registry, we store basically these different URI schemes. And then if you call our the SPARQL endpoint, you can get all the lists. So you can pass in the Perl URI, and then you can get all the different URI schemes that we have in our store. So if you don't have it, then you won't be able to do it. But then it's a matter of adding this URI into our registry.
Hi, Sarala.
Yeah.
This is Tom. I have a question about whether you're planning as part of the migration to cloud services to also migrate the triple store that you run, and if so, on what platform and how you're thinking about doing that.
So this is a virtual SPARQL endpoint. So it's actually not a triple store. Yeah.
Mm-hmm.
So we use I think it's Jenna. We use Jenna to convert these URI schemes. So, but we haven't really thought about moving this to the cloud. I would like to know who would be interested. If there's enough interest, yes, we could look into moving this into the cloud. So, I mean, there's really different views that I get about SPARQL and RDF. So we don't invest too much time on this. This was done for I mean, there was a community using it, and I haven't had any sort of problems with it so far. But in terms of RDF and SPARQL, there's a lot of division. So I would like to know more.
Yeah, no, this was a curiosity question.
Okay.
So I have another, I guess, less related question, which is about, I'm looking at the identifiers.org registry, and one of the things that we've had questions about with regard to RRID resolution is that you guys use pattern matching. And if I wanted to say, this pattern doesn't quite match, is that the file that I would submit a pull request to update how identifiers.org resolves RRIDs?
Oh, right. Okay. Yes, you could do a pull request on the registry, or you could, we should really streamline what we use, but I get requests from by email or as a pull request. So anything is good.
Okay.
I would like to, if there's some problem, it's good to get it done quickly. So.
Yes, it's just, I'm just staring there. It's not actually A to Z.
It's not clear, is it?
Yeah, it basically is. We will accept anything. Yeah.
Thank you. So I will make it clear. So let's stick with pull requests, because
Okay. Yeah.
Thank you.
You're welcome.
Well, excellent. Anita, please.
So I just I wanted to kind of highlight your role in this, and also the role of the individual repositories. So I noticed several times, actually, on your presentation, the word taxon and a colon, and then 9606, which is I believe human. I can't remember exactly.
You're correct.
Which is great. And so that would be human from NCBI taxonomy. Have you, but NCBI taxonomy has just updated how they would like to be referenced. And so I would like to kind of bring that up and say, you've put it on your presentation. They have a method that they would like to use. What is there, can you talk about how dynamic some of these things are, what the, what the backward compatibility looks like, and how do you make a change? What happens? I want my identifiers to look slightly different. What do I do? Right.
So okay, I wasn't aware of this one, so sorry about that. So in terms of the compact identifier scheme, that's kind of standard. That's what we're promoting. But how we access the data, the underlying URL can change, because that's what we store in our data repository. But because I don't know what they've changed it to, it's hard to kind of comment on it.
Yeah, maybe I could give a concrete example looking at.
Do you have a link?
Yeah, so for example, I think there's a balance that we talk about a lot, which is what we show people versus what we resolve. So, for example, I think the question that Anita was asking about legacy is about how do you have both the old taxon :9606, which is present and needs to be resolved in many places, but how do you also honor the desires of the folks at NCBI who maintain the taxonomy by saying, if you're going to use a compact identifier to refer to one of these taxonomy identifiers, please use this compact form. And do you make any distinction in that? Is there a place where you guys provide sort of the canonical or preferred compact representation for some of these, or how does that work?
So we don't really do that at the moment in terms of because I mean, for most of them, there's no preferred. Well, for ontologies, they do have a preferred way of doing things. So we are meant to add this information into our registry. But for other things, no. In terms of taxonomy, so let's say before we were using taxonomy :9606, and then people wanted to change it to taxon dot something :9606, and as long as the bit before the prefix, the colon changes, we could accommodate it. So if it's so tomorrow you want taxon : the identifier, we could resolve that as well as taxonomy : but if the format itself changes, but this is something that we are saying, you should use, it will be useful if everybody uses the same compact identifier format. So that's what we are suggesting. But in terms of how they, is it a URL change that they've done? Or is it?
No, in this case, it's the compact form that they would like people to use.
Oh wow! Okay.
Yeah, and so that, and the reason why I ask is because really, one of the things about compact identifiers, to me, seems that they were never intended originally to be globally unique. They were always intended to be accompanied by a set of prefixes that are that sort of tell you how to expand them again. And so what that means practically is that we have to continue to resolve all of the old compact forms, regardless of whether they're preferred or not. But the question is, and I think you answered this, you guys aren't really in the business of saying this is the preferred form. You're just saying we're going to resolve all of these.
Yeah. But it would be interesting. Yeah, I would need to read up on how they have changed, because this makes, need to think about that. Yes.
Yeah, we have to build some resolution on our end as well for their new preferred form, and because I think we don't yet resolve it, but I was wondering if you guys had, because I think we're on NCBI tax or
we conform to what OBO. We just use OBO. Right.
So, but we will have to continue to work with that version of it, and then but if they say something, they're kind of I think that no one else has taken their preferred form, so if that's their preferred form, then no one else has taken it.
So I guess in sort of as a summary, it's better to kind of show the preferred for forms as well. Okay. Right, I can add that. Because we will be doing that for sort of ontologies, but I wasn't aware of the other bits.
Yeah.
But thanks for this information. This is new to me.
Okay.
It was new to us too.
Yes, it was, but it's fine. It is. I mean, they've settled on something, and they are the authority. So, and it seems like from our side that it doesn't really interfere with anybody else. So, I think it's just, I think we also need to respect the wishes of the source, if they have any.
Yeah. Yes. Okay. Okay.
All right. Well, I think that might conclude our webinar. Sarala, do you have anything else you'd like to add at this juncture?
That's about it.
Excellent.
Thank you.
Well, thank you so much for joining us today. And I'd just like to give a quick plug for our next webinar on April 27th. We'll be joined by Dr. Molly Bogue, who will be talking about the Mouse Phenome Database, an integrative database and analysis suite for curated empirical phenotype data. Furthermore, I have recorded this webinar, and it will be on YouTube later today after I make some brief edits. Sarala, thank you again so much for joining us. We really appreciate you making the time at this late hour for you on a Friday evening. And I hope you have an excellent weekend.
Thank you so much for listening.
All right, I'm going to end the webinar now. Thanks again.
Thanks, Sarala.
Bye.